Electroforesis is a technique frequently used in the genetic engineering laboratory to know the size of DNA, RNA, and protein sequence. Each type of electrophoresis has correction conditions, a specific buffer, and the markers that allow a good performance in the application of the electric current. Knowing how to interpret the results is of great importance to determine the errors made and currently obtain the data of the size of desired bands.
Knowing the different devices and what they are for help us to work in the laboratory more efficiently. Electrophoresis is a technique that helps us to know some characteristics of different biomolecules. For example, it helps us to know the molar mass of proteins, DNA, and RNA in Daltons and base pairs. In addition, we can know the isoelectric point of proteins with 2D electrophoresis and verify amplification by PCR. Some of the applications that are given to electrophoresis are in criminology, examining samples of blood or tissue that are in a crime scene. It is also used in paternity tests, as well as establishing genetic links or visualizing bands of a molecular marker for genotype of individual plants. There are some aspects to consider for the realization of electrophoresis for RNA versus DNA. For RNA requires an extra procedure, denaturation before and during electrophoresis to prevent secondary structures that affect its migration are formed. For this, lyxyl DMSO, formaldehyde, and methylmercuric hydroxide are used. The denaturing gel should be washed to remove the denaturants before staining. Also use normally TBE buffer and use dyes with higher affinity for single-chain molecules that can increase the specificity and sensitivity in the detection of RNA. For DNA, you also can consider the large DNA fragments tend not to separate well in TBE buffer. For this, you can use TAE buffer. Also verify the percentage of agarose according to the sample to be used. For example, if you use genomic DNA, you can use 0.8% of agarose. And verify the appropriate voltage for an optimal shift. Similarly, there are variations of the electrophoresis technique for cases in which biomolecules cannot be separated by the conventional technique. The DNA molecules, which adopt a more or less globular conformation, have an excessively large size to cross the pore of the gel and do not separate depending on their size. For these cases, pulsed field electrophoresis is used, where 1% agarose gel are used, but the orientation of the applied electric field is altered periodically, activating alternately two pairs of electrodes. The frequent change of direction of the electric field causes the molecules to unfold and advance through the pores in extended conformation. A larger size reorient with more difficulty, so they move more slowly. This technique can be used for studies of genetic diversity. This item shows the elements to be used according to the sample. For a sample of plasmid PMO L28, we can use agarose since the plasmid measures 171,000 base pairs and the polyacrylamide matrix only serves to separate small fragments of DNA. The recommended buffer is type buffer because it works better with plasmids, and TBE is inhibitor of many enzymes. The loading buffer recommended is mixed buffer 6X, and the marker sample is the BAC tracker TM supercold DNA ladder marker because it's suitable for estimating the size of large supercold DNAs. The blending condition is 1 to 2 volts per centimeter depending on the distance of the electrodes. And the display of the sample is that we can see in the image that the plasmid PMOL28 is the second band above down.
The first step to follow for the preparation of the electrophoresis gel was to perform the corresponding calculations for agarose, buffer, and stain to obtain a 50 milliliters gel. Once the quantities to be used are known, prepare the indicated volume of type buffer and add the necessary amount of agarose. Dissolve the mixture in a microwave using insulating gloves to avoid burns, making sure it did not spill. Wait for the mixture to cool down, and before it gels, add the gel red 0.2x. Blend perfectly. Transfer it to the electrophoresis chamber mold and wait for it to gel. Now it's time to place the gel in the electrophoresis chamber and cover it with buffer. Add the appropriate amount of loading buffer in a piece of far film. Also add the required volume of the DNA sample and then mix DNA and loading buffer with a micropipette. In the first well, add 5 microliters of the molecular weight marker and then add the mixed samples in the indicated wells. Now, run the samples for 60 minutes and 100 volts. Using gloves, visualize the gel in a transilluminator. Finally, document the image.
In Figure 1, the electrophoresis of samples of DNA and RNA at different agarose concentrations using buffer TAE at a concentration of 1x as running buffer is observed. All gels were dyed with gel red at a 0.2x concentration and run at 100 volts during 60 minutes. The results of sample 1 were obtained in gel A, where the 11 bands are visible. As the concentration of agarose increases, a lower separation of the DNA fragments is observed. Samples 2 and 3 were not able to separate properly in the 3 gels. This may be because the length of the DNA fragments in close proximity had very small difference in size. The results of sample 4 in gel A display several distinguishable bands, whereas in gels B and C, the bands are smeared and at the top of the gel. The sample of genomic DNA is also smeared in the 3 gels. The RNA sample displays a smear at the bottom of the 3 gels, and a few bands at the top of the gel, but they are not well defined. Lastly, a unique band is shown in all 3 gels for the PCR sample, all indicating a size of 450 base pairs of the sample. In Figure 2, the electrophoresis were performed with the buffer TBE at a concentration of 1x as running buffer. The concentration of agarose is the same order as the previous gels. All gels were dyed with gel red at a concentration of 0.2x and ran at 100 volts during 60 minutes. Better results for sample 2 were obtained in gel A, where the 11 bands are visible. However, the smaller fragments have low visibility. In gels B and C, the bigger fragments are concentrated in the upper part of the gel, and the smaller fragments, just like in gel A, have low visibility. The DNA ladders in sample 1 and 3, the smaller fragments are distinguishable. The fragments bigger than 500 base pairs, since they are close together in size, do not separate properly. This is observed in the 3 gels. In the sample 4, different bands are observed in the 3 gels. The DNA sample is smeared. However, there is an intermediate band observed in the gels with a better resolution than it did in the TAE buffer. For the RNA sample, the different bands are the different types of RNA present in the sample and are better observed in gel A than in gel B and C. In gels B and C, these bands are not visible enough to be identified properly. Finally, the PCR product presents a good resolution on the 3 gels. In Figure 3, the following details of the electrophoresis with TAE as running buffer are identified. In blue, the different bands obtained from the plasmid are shown. These indicate the presence of different isoforms of the plasmid. And in lighter blue, the contamination of RNA in the sample is shown. In red, the degraded DNA is indicated. In green, the presence of different types of RNA in the sample are shown, and in yellow, the unique band of the PCR amplified is pointed out. Figure 4, the following details in the electrophoresis with TBE as running buffer are identified. In orange, the distortion of the DNA fragments of the sample is indicated. Just like in the previous figure, in blue, the different isoforms of the plasmid are visible. In lighter blue, the contamination of the sample with RNA is shown. In red, the degraded DNA is pointed out. In green, the different types of RNA in the samples are shown. And in yellow, the unique band of the PCR product is indicated.